Ever wondered how our bodies actually decide whether we become male or female? It's not as simple as just an X or Y chromosome. In today's video, we are breaking down the fascinating world of reproductive embryology. Let's dive in. For the first 6 weeks inside the womb, the developing embryo looks exactly the same regardless of its genetic sex. The anatomical structures that will become the reproductive system are identical at this stage. After the 6th week, the genetic instructions kick in, and the reproductive organs begin changing into either male or female structures. Sex of the conceptus determined by the sex-related gene, that is SRY gene, present in distal end of the short arm of Y chromosome. If the embryo has a Y chromosome carrying a functioning SRY gene, the undifferentiated gonads are instructed to develop into testes, making the fetus anatomically male. In the absence of a Y chromosome and thus no SRY gene, the default genetic path takes over, and the gonads develop into ovaries, making the fetus anatomically female. Let's clear some anomalies like Turner syndrome and Klinefelter syndrome. Turner syndrome occurs when a baby is born with only one X chromosome instead of two. Its genotype is 45X0. Because there is no Y chromosome, these babies develop as females. Klinefelter syndrome occurs when a male has an extra X chromosome with genotype 47XXY. Because a Y chromosome is present, the SRY gene triggers male development, so these babies are biologically male. This is how the sex of baby is decided. One most important thing is that, for proper development of ovaries, both the X chromosomes should be present, although both the X chromosomes are not needed for presence of ovary. Because of that, individuals with Turner syndrome 45X0 only have one X chromosome, their ovaries fail to develop fully. They degenerate early into non-functional, fibrous tissue known as streak gonads. The formation of the male gonads, testes, begins early under the influence of the SRY gene, right around 6 to 7 weeks of pregnancy. The SRY gene doesn't work alone. It activates a secondary, helper, gene called SOX9, which is absolutely critical for directing cells to form testes. The female gonads, ovaries, take slightly longer to begin forming, starting around 7 to 8 weeks of gestation. Even though female development is the default, it still requires specific genetic orchestration. WNT4 and RSPO1 are the key helper genes that promote ovarian development and suppress male pathways. Thanks for watching. Subscribe for more updates.